separate the different pieces of DNA from one another. And by the way, this is happening for each one of these cells. So this cell is going to go on and produce 2 cells, and this cell is going to go on and produce 2 cells. And so if I just made copies of this at the end of meiosis 2, you would end up with 4 cells, and the information we would have, because remember we had mom's DNA crossover with dad's, and then same thing, we had dad's that was crossed over with mom. So in meiosis 1, the first half of meiosis, you're separating the homologous chromosomes from one another. So in other words, this will be in this cell, and this one will be in a totally different cell. And so we have 4 unique strands of DNA. We have the recombinants, which are going to be the ones that change DNA. So at the end of meiosis 2, if we're separating the homologous chromosomes from one another in meiosis 1, then in meiosis 2, we're going to separate these sister chromatids from one another. So if we focus on separating this first, we're going to end up with a piece of DNA that looks like this in one gamete, so that's going to be this guy right here, the all purple guy. And then in the next one, you're going to have the recombinant piece of DNA, which is mostly purple, but has some from dad, or mostly mom, but has some from dad. And then if we focus on this one, same thing, you're going to end up one gamete that's all, all dad. And then the other one is going to be mostly dad with a little bit of mom tied into there. And so when you have these 4 different gametes, each one of them is going to be genetically unique. And so we'll have a piece that's all mom right here. We'll have a piece that's mostly mom and a little bit of dad. Here we have an all dad, and then here we have mostly dad with a little bit of mom genetics in there. And so these are all going to be haploid, or 1N, and they're all going to be totally genetically unique. And so if we look at the different possibilities of the ways that these homologous chromosomes line up in meiosis 1, remember we said they can line up randomly however they want. So you could have a mom there, a mom there, a mom there, and then something like that. And so since we have 23 different sets of chromosomes, there's 23 different ways, or there's more than 23 different ways, but there's all these possibilities for how each one of these chromosomes aligns compared to its homologous chromosome. And so the different number of possibilities of chromosomal arrangements is 2 to the 23rd power. So every time that meiosis occurs, this comes out to be, I think it's like 8 billion. So there's 8 billion different genetic combinations that can result in meiosis. And so this is how meiosis is one of the processes of introducing this genetic information. So depending on how the homologous chromosomes line up with one another, and depending on which parts of the DNA cross over with one another, and then depending on how they line up again in metaphase 2, can dictate the different genetic possibilities of all of these gametes. So much so that there are 8 different billion genetic possibilities. So when mom is undergoing meiosis to make her egg, and dad's undergoing meiosis to make his sperm, and then depending on which sperm fertilizes which egg, and again, each one is going to be genetically unique, because this could be, if this was a female, this could be one egg, this could be another egg, this could be another one, that could be another one. Same thing if this was a guy, that could be sperm 1, sperm 2. And so each sperm is going to be genetically different. And so depending on which sperm fertilizes which egg, you're going to end up with 8 billion different possibilities of genetics, which is why all of us look so very different from one another. And so if you had 8 billion kids, they would all look different. because there's 8 billion, I know it's a lot of kids, but there's 8 billion different possibilities of what your child's genetics would be, which means that it was going to affect, what your kid looks like anyways. So again, we're introducing this genetic diversity into meiosis. And so again, we're starting off, we have we have, after DNA replication, we have our homologous chromosomes, one from mom, one from dad. In prophase 1, you're going to see this crossing over between homologous chromosomes. And then you're going to go through the normal stages of cell division. So you're separating the homologous chromosomes from one another. So chromosome 1 from dad, chromosome 1 from mom. And you've already introduced this genetic variation through this crossing over in prophase 1. And then after you go into meiosis 2, there's not another DNA replication, except this time what we're doing is we're separating those sister chromatids from one another. So you can see you end up with these 4 unique gametes that are being produced. And then again, depending on which egg matches up with which sperm during fertilization, that's going to give you different genetic traits that are going to be found in that new offspring. Questions? Are y'all lost at this point? No?
So when during prophase 1, when they're crossing over, is that when like a recessive trait can come over you might just happen to get, or is that later on?
Yeah, exactly. So if there's a mutation, let's just say there's a mutation in this gene right here, whatever this gene is, and now you're giving it to the other one. So again, depending on where that mutation is right here, you could carry that into your offspring. So again, it all just depends on if that mutation gets, crossed over onto another chromosome or whatnot. And so crossing over is also going to be important because it can allow us to figure out where certain genes are. So if here's, let's just say this is my, dad's chromosome and here's mom's chromosome. Now, if some of these genes are going to cross over with one another, so let's just say I have an eye color gene right here. So this is eye color, and let's just say this is, hair color. And same thing on mom's. This is eye, this is hair. So depending on how far apart genes are on the same chromosome, that can dictate whether or not those genes will be separated from one another if they get crossed over. So in this case, because these guys are pretty far apart, it's a pretty good shot that you're going to expect them to switch places with one another through this crossing over. Whereas if I had 2 genes that were right next to each other, the likelihood is that you're not going to cross over in between those 2 genes, because the chances of splitting that directly are going to be very, very, very minimal. So the further 2 genes are apart from one another on one chromosome, the likelihood is that they're going to be separated from one another during this crossing over phase. And so by looking at the rate of crossing over or the rate of recombination, so recombination frequency looks at the rate at which this crossing over occurs. And so after crossing over occurs, again, you end up with these genetically unique gametes at the end. And so we have you're always going to have 2 recombinants. So this is a recombinant because it has both pieces of mom and dad's DNA, just like this is a recombinant. And then these guys are called parentals because they look just like the parents. They have a whole set of dads or a whole set of moms. And so at the end of meiosis, you're always going to have 2 recombinant gametes and 2 non-recombinant or parental gametes. And so again, you can measure this rate of recombination or this recombination frequency. And so if you have a recombination frequency of 50%, that means that these two genes are going to be separated from one another during this crossing over. And if you have a recombination frequency that's less than 50%, this means that the genes are going to be really close to one another like that, and that they're not going to be separating from one another. And so the way that you calculate the recombination frequency, you take the number of recombinants in an offspring and then divide it by the total, the number of recombinants plus the number of parentals. So it's just a percentage that you're taking. And so if this number is equal to 50%, that means that the genes that are found on these chromosomes are going to be separated from one another. Because they're so far apart on the chromosome, the likelihood that the crossing over is going to occur between them is at its maximum, at 50%. And then again, if you have less than a recombination frequency of less than 50%, that means that these genes are going to be so close to one another that they're not going to be separating from one another. And so when your recombination frequency is 50%, we say that the genes are assorting independently. So another way to say this is that they're following the law of independent assortment. And so if genes on a chromosome line up differently, and the genes are so close or so far apart that you know that you're going to be crossing over in between them, we say that they have been assorting independently. Now, if your recombination frequency is less than 50%, then that tells you that the genes are going to be linked to one another. And so when genes are linked to one another, that means they're going to be on the same chromosome as one another. And so just by looking at this recombination frequency, or the rate of crossing over, you can help use this to map genes on different chromosomes to determine if gene A is on the same chromosome as gene B, or if gene A and gene B are right next to one another on the same chromosome. And so you can calculate that just by looking at your offspring. After you've made your gametes and those gametes fuse to make offspring, you can look at the genetics of them and determine if they're recombinant, if their DNA has been changed between mom and dad, or if they look just like the parents, meaning no recombination, no crossing over occurred. Okay. Questions? Yes, go ahead.
So is eye color always going to be on say chromosome 2?
Yeah.
I was researching this because I missed a class, and earwax, random, is always on chromosome number 16. And whether it's dry or wet is just by one place of DNA.
Yeah, and so yeah, exactly. So the length, we have our chromosome right here. Let's just say this is chromosome 1, and I have no idea which genes are on chromosome 1, but let's just say, gene A, gene B, gene C, D, E, so forth and so on. And so it doesn't matter if that's mom's chromosome and this is dad's chromosome, the same genes are still going to be there. Again, they just might be different versions of that gene. So eye color, hair color, some of the other simple genetic traits where it's only one gene involved. And a lot of these traits that we talk about, like eye color, there's just one gene that controls eye color. While other traits like, the height you are or your pigmentation of your skin, those are going to be more complicated and regulated by several different genes. So these traits can be real simple and can be affected just in one gene, or it takes multiple genes that need to be expressed in order to see, the phenotype, to see the outcome, how tall you are, the color of your skin, so forth and so on. What?
That's what I promised.
Okay. I want to, just because we're here and we've already got this going, I want to, let's see, search for maybe a video animation so y'all can kind of see this in action, because I know drawing X's on the whiteboard probably isn't the most efficient way. Let's try this. Hopefully we get some volume.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis I, halves the number of chromosomes, and in the second division, meiosis II, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase I, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only 1 member of the homologous pair and will be haploid. In telophase I, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only 1 of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
That help at all? Maybe a little bit? Okay. And so, one thing, if you ever want to see any of the animations of any of these processes, McGraw-Hill is a good place to look because they actually you know, have these videos and stuff that animate and give you a pretty good rundown of what's going on. Okay, so that's meiosis. And like I said, I'll get a PowerPoint slide up, and then I'll record the next lecture, which is tomorrow morning. It'll be the same lecture, and so it'll just be a little bit more professional looking than this one. Yeah, Lindsay.
Okay, so you know, rough draft is due this Friday for the
Yeah.
lab report.
Yes.
Are you gonna post something on how to do it?
There's already instructions on Blackboard that say how to do it. So there should be a folder that says lab folder, our lab report, and then it's got instructions, or y'all know? No?
I only saw what was in the lab 5.
All right, well, let's just look. So,
Do you need help?
and we did talk about it in lab, how to kind of set it up a little bit.
Do you have school?